My name is Suzanne George. I'm a medical oncologist here at Dana-Farber in the Sarcoma Center. And as others have mentioned, we see many different types of sarcomas, and today's focus is really on GIST, but I will make some comments as relevant to other sarcoma types as well. Let me see. So primarily what I'm going to talk about today has to do with advances in systemic therapy, so drug therapy. Dr. Rout did a great job talking about local therapy with surgery, and now we're going to shift gears and talk more about the drug therapies that we use. We are going to talk a lot about Sorafenib and Regorafenib, but I do want to put it into some background. As many of you know, the mainstays of therapy for the management of advanced GIST is Imatinib for first-line therapy and sunitinib for second-line therapy. These are the only two drugs that are currently approved by the U.S. FDA for the management of GIST. And these two treatments have absolutely revolutionized the way that we treat people with advanced GIST, and are now even going earlier into the disease in some patients where we feel it may be appropriate to use the therapy after resection of just one lesion of the primary tumor. But despite the tremendous advances that we've made with regard to the management of this disease with these drugs, resistance can still be a really stubborn challenge for many patients. And this is an area that we clearly need to continue to do hard work and to get better options moving forward. So as you've probably noticed as you read about GIST and learn about GIST, that most of our drug therapies end in IB, and they are tyrosine kinase inhibitors. These are drugs that sort of target in on specific proteins that are thought to be important in the biology and the pathophysiology of this disease. And many of these tumors share very similar characteristics, but in many ways they also differ. This diagram actually, pointer here, it works. What this slide shows is this circle is Imatinib, and what it shows is that these are the targets of Imatinib. Imatinib is a relatively narrow spectrum kinase inhibitor. It blocks a relatively few number of protein targets, but it does it really, really well. This is sunitinib, also blocks a number of proteins, but has somewhat of a broader spectrum than Imatinib, and also hits a number of targets that are different than what Imatinib hits. What you can see here is that Imatinib and sunitinib share some common targets that they block. And what they share are blocking KIT and blocking the platelet-derived growth factor receptors alpha and beta. And it's really these targets here that are so critically important in the biology of GIST, and it is so critically important in the management of GIST. And what we know is that even though they both carry these similarities, there are some differences, and what these differences may be related in part to some of the side effects that people experience, and also to some of the different diseases that you may see these drugs indicated for. What's also important to note is that even though these drugs are very similar, they are different in their structure and in their spectrum, as I mentioned. And when one IB stops working, then another one may pick up, may be effective in that situation because of these differences. This is a complicated slide, and really what I want you to focus on is just the area in this oval. And what this is showing is that for some patients who develop a resistance mutation to Imatinib or Gleevec, sunitinib may actually work. And what these little squares show us is that these are specific mutations where the red means Imatinib may not be is likely not effective, but some of them, sunitinib may similarly not be effective, but sunitinib may actually be effective, and it may be in part not only related, likely not completely related to the spectrum of inhibition, but how the drugs actually block the target. So as we start thinking about where we go beyond sunitinib if additional resistance develops, where else do we look? And many of the studies that have been done to date and many drugs that are being evaluated are also part of this family of multi-targeted kinase inhibitors. There's dasatinib, nilotinib, and pazopanib. These are all drugs in this same class that are approved for other diseases, not for GIST, but that are being looked at in GIST, but approved for other types of cancer. And then, in addition to these approved drugs that are being explored in GIST, there are actually investigational drugs, Imatinib and Regorafenib, that are in the process of being evaluated in patients with GIST. I'm going to focus primarily on Sorafenib and Regorafenib. So Sorafenib, as I mentioned, is another broad-spectrum kinase inhibitor. It's similar to sunitinib in its spectrum, meaning in terms of the targets that it hits. It's closer to that big circle that was on the sunitinib side. It hits KIT, it hits platelet-derived growth factor receptor, alpha and beta. It hits a number of VEGF targets. It hits RET. It's similar. And we also know from some preclinical data that was done by Dr. Fletcher's lab as well as Dr. Heinrich's lab that although they're similar in their inhibition, that they do block, that Sorafenib blocks some mutations that are not blocked by either Imatinib or sunitinib. It's currently FDA approved for the use of metastatic renal cell cancer. And in that patient population, so in people that have metastatic renal cancer, it has been shown that let me back up a second, say metastatic renal cell cancer is a disease that's standardly treated with sunitinib. It's the other indication other than GIST for that agent. And what's been shown is that people that have metastatic renal cell who are treated with sunitinib, but then sunitinib isn't the right fit anymore, the disease starts to figure out a way to grow despite it, that Sorafenib can be effective. So what that can do is it implies that there are some overlapping but biologic distinctive effects in patients. These drugs are similar to each other, but when you actually get them into the patient system, they work differently. And that allows us potentially more opportunity for more agents. Let's see. So what do we know about Sorafenib and GIST? There's been one phase II trial in the United States that evaluated Sorafenib in GIST, and these were in patients, all patients were treated, had been treated initially with Imatinib, where Imatinib resistance had developed, and some of the patients also had been treated with sunitinib. And in this mixed population of Imatinib and sunitinib treated GIST patients, most patients that received Sorafenib demonstrated disease stability for some period of time. There was a European study that actually looked at this agent as fourth-line therapy. So after receiving Imatinib, SU, which is sunitinib, or nilotinib, so these patients had all received 3 prior lines of therapy for GIST, and Sorafenib was then administered. And very similar results were found. Most patients, the tumor would stop growing for a period of time. The challenge always is, how long will that period of time last? And in some people, it was a number of months, a relatively short number, and in others it was longer. On average, it was about 5 to 6 months in actually both of these trials. So what that tells us is that Sorafenib has activity in this disease, but there may be an opportunity to do even better to see if there are drugs that may continue to work in this patient population, perhaps somewhat more durably. So we've continued to investigate other drugs in this class. And that brings us to a new drug called Regorafenib that is investigational, has not been approved yet for any indication in the U.S., and remains in a number of clinical trials. When this drug was being developed, just based on the structure, its shape, it was initially known as fluorosorafenib because it's so similar to sorafenib in some ways in its structure, but it's also very different. There's one area that's just shaped just a little bit differently, so that even though it hits a number of the same targets as sorafenib, it may do so in just a slightly different way, so that the spectrum is just so slightly different. And its early data using this agent suggests that it is quite an active drug in renal cell cancer. Again, renal cell cancer is a drug that sunitinib is active in and that sorafenib is active in. And so regorafenib as an active drug in this disease is suggesting that it has that it can hit the targets that are relevant, and it seems to be doing so in a promising way. Because it showed such promising activity in renal cell, when similar drugs had been used as we use in GIST, we then went on to study it in GIST. Now, there's a phase II trial that we ran from here, multicenter, evaluating Regorafenib and GIST. Dr. Butrinsky is going to talk a little bit more about the different phases of trial to kind of to put some context around that. But briefly, a phase II trial is a study where everybody receives the drug, everybody receives the same dose of the drug to start, and the patients enrolled in the trial generally have the same disease. We have completed enrollment in this study. 34 patients have been enrolled to date, and this was a study that completed its accrual in about 9 to 10 months. Regorafenib is a drug that's taken orally, the same as the other kinase inhibitors that we use, and it's on an intermittent dosing schedule, so it's given for 3 weeks in a row and then is followed by a 1-week washout. Some of the really important things that we thought when we were designing the study were critical to include were not only obviously following patients in terms of their scans to see what happened to their tumor in response to this drug, but it was very critical to get tumor tissue available for analysis. And so we could understand some of the subtle differences that we all know exist in GIST and to see if that related at all to how patients responded to this drug. And the tumor that we collected was not only archived tissue, meaning tumor from prior surgeries, but in a subset of patients who opted to allow consent for new biopsies, we were able to gain fresh tissue to look at the current status of mutations and to see how that may impact the potential effectiveness of this drug in this patient population. We also did PET scans extensively as this study, really as a research tool to help us understand metabolically how the tumor may be changing in response to the agent. And we did blood level testing, and also a number of samples were taken of urine and blood so that we could see if there were any biomarkers, meaning substances in the blood and the urine that may be related to response or that can tell us how the drug is actually hitting the targets that we believe that it should be. So that phase II trial, the results are not yet mature enough to report, but we will be reporting them at the American Society of Clinical Oncology meeting next month, and that will be our preliminary evaluation. The Regorafenib, though, has shown promise in a number of clinical systems, as I've mentioned, in a phase III trial of this agent in gastrointestinal stromal tumor in GIST is ongoing. And I want to talk a little bit about the phase III trial design. This is a randomized, double-blind phase III trial. And what that means is, in contrast to the phase II trial, where you enroll a relatively small number of patients, everybody gets the drug, starts at the same dose, and then are followed, what a randomized phase III trial does is the computer decides. You either get chosen, the computer either randomizes you to one arm or to the other arm. And in this study, the arms are either Regorafenib or placebo. And placebo is a pill that doesn't have any active drug therapy in it. It's a sugar pill. And then patients are followed very closely, initially every 2 weeks for side effects. And the scans are done every month initially to evaluate any potential change in the tumor. What this study allows is that if there is notable growth in a patient, then it's determined what arm the patient is on. And if the patient was on the placebo arm, then the patients can rapidly be switched over to Regorafenib if they choose, together with their study team, and then continue on the study. Patients that are eligible for this trial are people with metastatic GIST. People must have received prior Imatinib and prior sunitinib. But prior treatment with Sorafenib is not allowed on this study. It would mean it wouldn't be the right fit for enrollment. This study is designed to open in 85 countries worldwide with a goal accrual of just under 200 patients. And there are several centers in the U.S. that are opening. We are open. We were the first center to open. Fox Chase will be opening if it hasn't opened already, and a number of other sites in the U.S. will open soon. This study design is designed as a registration trial, one to definitively identify whether this drug has adequate activity in this disease to lead to regulatory improvement, regulatory approval to allow access to the drug to patients wherever they may be, not just limited to people who are seen in subspecialty trials or enrolled on clinical trials. So in summary, broad-spectrum kinase inhibitors continue to show promise in advanced GIST following therapy with Imatinib and sunitinib. These are drugs that have overlapping characteristics but also have distinct properties as well.